The total number of samples collected were 18, 3 chickens from each area. Samples were gathered from 2 areas, and 3 different chicken parts were swab, skin, cloaca and tongue, and esophagus. After obtaining the samples from the field and stored in the incubator, the next step was to perform the serial dilution. Serial dilution is the process conducted to determine the colony-forming unit or the actual amount of bacteria present in the sample gathered from the field. It is done by placing 9 milliliters of distilled water in 5 test tubes. 1 ml from the sample will be taken and transferred to the first test tube. Flame the test tube before transferring the sample. 1 ml of the liquid will be taken from the first test tube and transferred to the next test tube. Shake the test tube well. The same process will be observed until it reaches the 5th test tube. The 5th test tube contains 9 milliliters of distilled water and 1 milliliter from the previous test tube, which will be used in the streaking. After serial dilution is carried out, streaking will follow. Agar will be prepared for the streaking of the sample. The agar prepared were potato dextrose agar, McConkey agar, triple sugar iron, brain heart infusion, and PPLO agar, and Salmonella shigella agar. The amount of agar to be prepared is dependent on the number of samples collected from the field. The agar was weighed and cooked for 1 minute. 10 milliliters of the prepared agar was transferred to the petri dishes. The petri dishes together with test tubes were placed in the autoclave for 20 minutes. After 20 minutes, the agar was removed from the autoclave, placed in the biosafety cabinet. Remove the lid of the petri dish to let the agar cool. Make sure to place the petri dish on a flat surface. For PPLO agar, 3.5 milliliters of Mycoplasma enrichment was added to 10 milliliters of agar. After the agar was cooled, streaking will begin. 4 samples will be streaked to 1 petri dish. The inoculating loop will be flamed before dipping into the sample. The dipped inoculating loop will be streaked to the petri dish. After streaking all the samples, the petri dish will be placed in a corresponding incubator for growth and culture. The samples streak in PPLO and BHI agar will be placed in ACO2 incubator and the rest will be placed in the aerobic incubator. Quantifying of colony forming unit and bacteria structure and growth. After 24 hours, the culture sample will be checked for growth and development. Identified bacterial and fungal growth will be subject to quantification and qualitative data. The number of colony-forming units will be observed and the characteristics of the bacteria such as margin, elevation, color, texture, shape, and size. The identified bacteria will be subjected to broth preservation. In preparation of broth for storage, the number of identified colonies of bacteria will be the basis of the amount of broth to be prepared. Bacterial that are isolated will be transferred to the broth that corresponds to the agar where it was cultured. There are available broths for PDA, PPLO, and MCA, and for bacteria from SSA, BHI, and TSI agar will be placed to peptone and MHB, which are used for general propagation. Peptone water is used in the identification of indoles such as E. coli. The broth is autoclaved and cooled. A small portion of the identified bacteria will be isolated using an inoculating loop. The isolated bacteria will be transferred to the broth and stored in the corresponding incubator for 24 hours. After 24 hours, the broth will be subject to observation as to change color and turbidity. For peptone, it was added with 5 drops of Kovacs reagent. The formation of a red/pink ring at the top of the liquid signifies an indole bacteria such as E. coli. The stored will be used in streaking. 1 petri dish will be utilized for a single bacteria. The same streaking process will be observed. The petri dish will be stored in the respective incubator for 24 hours. After 24 hours, the growth of bacteria will be observed. If the bacteria that grow is the same as the previously identified bacteria or the expected growth, it will undergo gram staining and pure culture. The data collected will be summarized and put in a document.